Welcome to the AI Institute for Next Generation Food Systems Fall Speaker Series. My name is Hannah Bartram, and I am the AIFS Education and Public Engagement Coordinator. Today, we are delighted to be welcoming Professor Gail Taylor, discussing AI challenges for fast development of high nutrition crops. A quick introduction to the AI Institute for Next Generation Food Systems, also known as AIFS or AIFS. The Institute was established last year under an NSF USDA NIFA grant with the mission of developing and leveraging transformative AI for the ethical production and distribution of safe, sustainable, nutritious food with fewer resources. AIFS is a multidisciplinary collaboration between researchers at 6 different institutions conducting research with a holistic integrated view of the food system, solving challenges in molecular breeding, agricultural production, food processing and distribution, and nutrition, and also focusing on the people behind the food system, using AI and technology as an enabler. If you would like more information on the research and engagement activities of AIFS, please visit our website and connect with us on social media. Our speaker series features thought leaders in addressing food system challenges using artificial intelligence. We hope you will join us again for these future webinars, and you can register for these events at the link at the bottom of your screen. The recordings from our speaker series will be posted to our YouTube channel. Today, we are delighted to welcome our second speaker. Gail Taylor is a distinguished professor and chair of the Department of Plant Sciences. The department is home to 71 professors, and key research thrusts in plant sciences include plant breeding, genetics and genomics, crop produce quality and safety, and environmental informatics. She holds the John B. Orr Chair in Environmental Plant Sciences, and her research interests include sustainable food production systems, including breeding for enhanced quality in leafy greens. She is also a member of the molecular breeding cluster of AIFS. There will be time for questions at the end, so I would encourage you to submit your questions throughout the talk in the Q&A section, and there is also closed captioning available today. Welcome, Gail. Take it away.
Thank you so much, Hannah. Let me share my screen. There we go. Just trying to hide the meeting controls. Great. Thank you, everyone, for joining today. It's a pleasure to be here. I'd like to talk to you about some of the AI challenges for fast development of high nutrition crops. And I'm here alongside some of my colleagues from the Department of Plant Sciences, Christine Diepenbrock, Dan Runcie, Alan Van Dyne, Charlie Brummer, Richard Mitchellmore, and Paul Gepps, and they have given me some valuable insights and help in putting this presentation together today. They're a wonderful group of experts. Please call on them if you'd like further details or insights. If there are any mistakes in the talk, they're definitely all mine. So I was asked to think about the challenges for crop nutrition and how we can move forward with AI. And I could really identify 4 key challenges which we might consider in a little more detail today. And they're shown here. So challenge number 1, crop nutrition and quality have traditionally not been a priority in plant breeding. So getting enhanced nutrition crops really has not been top of the agenda, at least until about 20 years ago. The second challenge, traditional plant breeding, which is what we need to get enhanced nutrition in food, is slow. The third challenge, plant breeding is extremely complex, and plant breeders wrestle with the plant genotype, the DNA that makes up the genotype, the plant phenotype, a multitude of characteristics which are important in determining the trait of interest. And at the same time, the environment, the rainfall, the soil type, etc., and time, how things change year on year, can also impact our outcomes. And then finally, challenge 4, slow may prevent us being fast. What does that mean? Well, slow in this context means social license to operate, in that we have to always be cognizant of the fact that the public, people out there generally, may not be accepting of all of the technologies that we have on offer and can be deployed to address this question. And as scientists, we always need to keep hold of that and remember it. So why are we interested in this particular topic? Well, this is the wonderful world of nutrigenomics and nutrigenetics. And along with many other people, I'm not quite sure what I understand by these terms, but what I think I understand is that individuals over here can have different gene expression responses to food and different patterns of gene expression. They have different metabolotypes, and that when they eat nutritious food, this can have a different impact on the individual. And at the same time, there can be quite far-reaching impacts which can actually modify through the epigenome the genome, the functioning of the genome of individuals. And so in the future, we need to grasp this, understand it, manipulate it, work with it. And the human population, or many of the human population, are really getting to grips with some of these ideas. However, central to this, and the domain in which I work, is the food stuff, and particularly plants, which is what I am particularly interested in. And this, I hope I'll be able to show you today, is a huge bottleneck for this area of science. So understanding plant nutrition, how it impacts human individuals, and the consequences of that for human health are still to be unraveled. And we heard some of that in our last talk. But the point I'd like to make today is that just modifying plant foodstuffs is extremely difficult and in some way in its infancy, and we need to accelerate progress in this area. So why is plant breeding a bottleneck, and how can AI unlock this food plant bottleneck? Well, we can define, for those of you who are not plant breeders, we can define plant breeding as the genetic improvement of plants for human benefit. And that was one of the many definitions given by the National Association of Plant Breeders. So we want to change and improve plants for the benefit of humans. This is a very, very long activity which has been going on for at least 10,000 years and probably more. And this unique relationship between people and plants began when early settlers realized that they could stop seed shatter in some of the early crops, probably wheat was one of them, and gather the seed to make flour and then to resow the crop. So this was the very, very early beginnings of plant selection and breeding. So this went on for some considerable time, that farmers were selecting the best seeds and growing them back each year in farming. And not until 1865, when Mendel's laws of genetics came along, did we begin to be able to control that process by making controlled or developing crosses, because Mendel, without understanding anything about DNA, was able to find these discrete entities through which inheritance occurred. And so crossbreeding was formed, and then after that, hybrid breeding, where different individuals were crossed to make very, very superior crops. And a good example of that is sweet corn or maize, that the hybrid, through a process called heterosis, is much more vigorous than either of the original parents. After that, in the 1940s, scientists used how to use mutagenesis, so through chemicals and other means, to introduce more genetic variation that could be utilized in breeding. And then finally, in 1953, Watson Crick and Rosalind Franklin discovered the structure of the double helix of DNA. And that was the beginning of how genetic information was really used to inform plant breeding. And crosses were much more directed. And then eventually in the 1990s, tissue culture led to the introduction of foreign DNA into plants, so the first GMOs. then the use of single markers in DNA to generate new plant types in the early noughties in 2000. And this is important because this figure is a little bit out of date, but a new type of marker-assisted selection called genomic selection has very recently come to the fore, and I'll come back to that later. It's a way we can accelerate plant breeding for the future. And then finally, we also have a much more targeted molecular scissors in the form of gene editing. So this journey of plant breeding, which has been going on for at least 10,000 years, has taken us to where we are today. So the first challenge then that I highlighted is that crop yield and not crop nutrition and quality has been the focus of plant breeding, certainly for the last century. So I've taken that figure on the left from an old paper that was published in the 60s, and it's describing the idiotype for yield in a particular grain crop. And it describes the characteristics that breeders were looking for at that time. There was a pressing need to increase yield and to feed the world, and this was really important. So these idiotypes for yield were defined, and breeders have been incredibly successful. So in general, yield has been enhanced by 1% per year, so about 10% per decade. And you may have seen these types of figures before. It's for corn yield, and it just shows the dramatic power of breeding over the last century to increase the yield of multiple food crops, in this instance corn. So there's very much a focus on the calorific value of food, and we were incredibly successful. And later on, scientists have focused on disease resistance and then abiotic stress resilience. But nutrition and quality until about 20 years ago was way down the priority list. The overriding effect of this domestication and breeding in general is that we've produced on the right here fantastic high-yielding food crops, but genetic diversity, if you think of all of these circles as representing a gene, over this 10,000 years has probably declined in our commercial breeding lines, such that we have less diverse germplasm in which to work at the moment. And that's a known factor that we need to address as we move forward. One of the consequences of that is that plant breeding has been phenomenally good for yield, but we've probably bred out many important phytonutrients which are really useful to human health. So for example, this is for total phytonutrients for apple juice. And you can see the wild crab apple had much higher values of phytonutrients than some of the modern varieties that we all eat. And we're all sort of intrinsically familiar with this phenomenon that perhaps flavor and nutritional value, something that we value in these foods, has changed with breeding. It also happens in my favorite topic, which is leafy greens. So if you ate a dandelion, taraxacum, it would have much more antioxidant power. So leafy greens, the antioxidants tend to be polyphenolics and other chemicals, but the iceberg lettuce, well, that's not very good in terms of the phytonutrients. So it's happened on multiple occasions, and these are some of my own data from my own laboratory, and we're comparing here a trace for the chemicals in wild lettuce on the top here, and which is a lactuca, a wild lettuce, and a bred lettuce, which is an iceberg type. And we were interested in the antioxidant potential of lettuce. And you can see here that we became interested particularly in quercetin. It contributes to antioxidant potential, and I'm not going to talk about this in detail today, but we developed a molecular marker for quercetin, and we bred some lines of high antioxidant lettuce. So we bred a lettuce with enhanced nutritional value. So we can exploit this wild lettuce, perhaps, for improving nutrition. But the point I wanted to make about this figure is that look at the trace here. In the wild lettuce, there's a more complex chemical signature, which is a chemical signature perhaps for nutritional value and we don't really understand necessarily what this signature means. So AI can help us in defining what we mean by a high nutrition crop or variety. Is it clear what high nutrition means? Can AI help us identify which combinations of the many micro and macronutrients are most useful? And this is a comment made by Dan Runcie to me, and this trace really exemplifies that. We might know what the knowns are, quercetin in this case, but what about these unknowns and how they impact on human health? That's a challenge. However, despite that exciting challenge, I wanted to make the point, this is Richard Mitchell here, took over a decade, about 15 years, to develop the resource that enabled us to identify the genetic marker for antioxidant power in lettuce. So it was Richard who made the original cross between the wild spiky lettuce here, Lactucas areola, and the iceberg type to make this mapping population, which we tested and we still test in multiple environments most years. So we have over a decade of data on this. And it just gives some insight into one of the stresses and strains of plant breeding. Nevertheless, as we move away from yield, in the last 20 years, some progress has been made, and that food crop nutrients are now being improved. This is taken from a paper in 2018, and it highlights 3 routes to this improvement. The first is breeding, so we can keep breeding, crossing, selecting, and making improvements. The second is using transgenics or editing now. We have very precise ways to edit plants. And then the third is crop agronomy management. By managing the soil and the conditions, we can also improve quality. So for example, you can see golden rice here, vitamin A rice, which was improved through a transgenic route, and mangoes and sweet potatoes have been improved. We have the orange sweet potatoes. So, but the point I wanted to make, so 20 years, a couple of decades, yes, we're making progress, but I still wonder if these are just the known knowns. We're doing zinc and vitamin A and the anthocyanins. I should have pointed out tomato here, purple tomatoes packed full of anthocyanins. We are making progress, and these things are coming through the breeding pipeline, so we should be aware of that. It's never been more important than it is today. So this document was published a couple of years ago now, where World Wildlife Fund and NOR got together with a group of experts to identify future 50 foods, which were selected on the basis of their high nutritional value, relative environmental impact, flavor, accessibility, acceptability, and affordability. And this is a great read if anyone hasn't read it. And these are the lists of the categories of food that they came up with. So some cereals and grains, as you might expect, but they're not the traditional ones. Lots of fruits and vegetables, and high value foods for the future where we should be focused. And most of these are where we know the very least about how we breed and improve for nutrition. So here's an example of that. This is from my colleague Alan Van Dyne in the Department of Plant Sciences, and he's one of the leaders of the African Orphan Crops Consortium to alleviate stunting due to malnutrition in African orphan crops. So these fit very well with those future foods, in that they're the crops that they're not the staples that are used across the world at the moment. So there's a 2020 report by the Lancet. Actually, there have been several of these reports that really highlight that people in the world are malnourished, that 1 in 4, 150 million children are physically, mentally, and economically disadvantaged, and poor diets and malnutrition are a key part of that. And this isn't through personal choice. Many people in the world still cannot access or afford a healthy diet or high nutrient food. And this is very pressing on the African continent, and it's where the Orphan Crops Consortium operates. And they have identified investment in nutrition and a role for plant breeding as being central to take us forward. and identifying some of these non-traditional crops and how we need to invest in them. So, for example, this shows that the aims of the African Orphan Crop Consortium are to end malnutrition and stunting. But there's a focus on these traditional, highly nutritious African food crops. And I wonder if this is another way that a place where AI can enable fast progress to be made. They're sequencing 101 genomes of some species that don't look familiar, but they're usually highly nutritious and have value to local communities in Africa. And this is a big consortium with lots of involvement of academia and industry. And it's fantastic to see this moving forward. But it does highlight a point which moves me on to my second challenge, which is that plant breeding, traditional plant breeding, is very slow. So this is the central dogma of plant breeding, that we have genetic variation. We're always trying to increase that variation in some way. We select plants through multiple means, as we saw on the timeline. We make crosses and we keep selecting, and then new varieties spill out of this testing pipeline. And it's a very slow process. Well, how slow is it for those of you who are not plant breeders? It's slow. Plant breeders have to be in it for the long term traditionally. So they may start up here in year one, where they make some crosses and hybridizations through parental selections. Before that, they have to get those parental selections, but let's start here. And then they might take 3 to 5 years to what we call fix the trait. So introgress that trait into the lines of interest so that it has some chance of being robust. And then 2 to 3 or more would be in field trials in multiple locations to ensure that what they think is happening is happening. And then the very best breeding lines would go to a national test centre to see if the new variety really is new, it's distinctive and can be characterised as something new. And this takes at least, at a minimum, 2 years. And so you see very quickly, it takes a decade traditionally to get a new variety of any food into the fields and into the supermarket so we can all purchase it. So we need new, faster technologies, and we do have some of those, and they're where AI can be of benefit. And I've listed 3 here: speed or speedy breeding, genomic selection, and gene editing. And genomic selection and speed breeding can particularly benefit from AI. I won't have time to go into many details on this today, but you're very welcome to call on my colleagues who are the real experts. So that's the second challenge. Plant breeding traditionally is slow, but we've figured out ways of speeding it up. AI is central to enabling that to deliver nutritious crops. What about the third challenge? This is really about complexity. Plant breeding is complex, and plant breeders wrestle with the plant genotype, the plant phenotype, the environment, and time. And all of these are multidimensional data which which we need to cohere together to move the breeding pipeline forward. So one way of putting this is it's a numbers game, or really it's a maths game that we can tackle. And we need to put the numbers together to make predictions to speed up the pipelines. And of course, AI can help us to do that. Plant breeders are always trying to find these favorable combinations of phenotype and genotype that are robust in time and space. And AI can improve these models of phenotype and also genetic background, and the importance of multiple genetic markers to move us forward. There's a general comment that genotyping, the genotyping part of this process, has become relatively cheap, but the phenotyping remains relatively expensive and requires more labour. And AI can help to find cheap and easy phenotypes, or remove the need for phenotyping altogether in more and more of the pipeline. So by genotype, we mean the DNA sequence that underpins breeding. So it's the sequence of every individual in our breeding population. And the phenotype can be any characteristic. So it might be the rate of an enzyme working. It might be the rate of gene expression of a particular gene. It might be the way groups of genes work together. It might be a morphological phenotype. It might be an adaptive phenotype to the season or a particular environment. So plant phenotypes are very, very complex. How do breeders deploy all of this together in a breeding program, and how do they do that to move forward? So this is again some work from Alan Van Dyne's lab. It's for a leafy green, one of these orphan leafy greens, and it's a roadmap for a breeding program, and it really lays out all the steps that need to be taken. Starting up here, we really need to understand what it is the target for breeding. So for example, I work in a leafy green, watercress, and we know that a target there is glucosinolates. Why? Because glucosinolate chemistry is known to have an impact, a positive impact on human health. However, glucosinolates are quite bitter, and in general, one of the reasons that health-giving chemicals have been bred out of modern crops is that they impart a bitter flavor, and humans have selected for sweetness. And so we're trying to introduce a flat, sweet flavor with that important bitter chemical. But you need input to understand what is the target for breeding. Then the breeder goes about getting germplasm, the most diverse germplasm, the germplasm of local importance adapted to the local climate. It might be from a gene bank or from collections or from other breeding programs. So again, more definition of the trait. How do we measure the things that we're interested in? Phytonutrient content. So in my case, it might be measure the glucosinolates, measure the sugars, but it might be something about shelf life or organoleptics, mouthfeel. And then other key characteristics of the trait. And then through this iterative process of getting the genomic data, where we detect variation in single nucleotide polymorphisms, for example, the single bases in the sequence of the DNA, and through deploying these genetic tools, these mapping populations and different combinations of crosses, we can unravel where this phenotype sits in the genome and begin to select for it. The traits are tested in multiple environments, a value chain assessment is done, and eventually we get some new genotypes being released. Another way of putting that was given to me by Paul Gepps, a colleague in my department, who sees huge potential for AI in breeding. And he's interested in high protein crops, phaseolus beans, the things, some of the beans which are going into meat substitutes significantly just now. And Paul at the moment is breeding for stress tolerance to make his beans tolerate low inputs, which in this case are irrigation. So over here, this is what the genotype might look like for a breeder on the right here. It's the chromosomes here of bean, and each of these dots is a molecular site or a marker that correlates to a trait of interest. So Paul and his team have been able to isolate or locate these areas of the genome that are of interest to them. And these are the traits of interest, and we call them these areas QTL, quantitative trait loci. They can occur differentially at different stages of growth and development and in different genetic crosses. So this in itself, unraveling this, is extremely complicated. But then we move over to this complexity, and this is a little bit what it looks like. So as a breeder, as we've said, we'll be testing these combinations in multiple years at multiple locations and in multiple treatments. And so you have this complexity spreading out here of these treatments. And then Paul's interested in growing these beans in water restricted conditions. And it turns out there's not a single gene for that. It's what we call a quantitative trait, and he needs to be assessing all of these phenotypic traits in his multiple sites. And some of them are to do with growth vigor, and these look particularly important. But the root traits also appear to be important. And overlaying all that, he has the environment. So the climate and the soil, which is impacting how he takes the breeding pipeline forward. So the outcomes are traits for selection combined with his genotype information. So I hope that's given you some insight into the complexity of some of the things that breeders are trying to wrestle with, and how AI may be able to accelerate this pipeline and enable us to make faster progress. So again, this is the decade to produce a new genotype. So I wanted to spend the last part of the talk really going into a little bit more detail on approaches we might be able to use, particularly in genomic selection. Why has genomic breeding or genomic selection become so important in the last decade or so? Well, it really relies on this, that in about 2006, actually, a new technology for sequencing DNA was developed. And this led to a huge tumble in the cost of sequencing any genome. So a project I was involved in at this time, it was about 2002, we spent $35 million to sequence the genome of one tree. And that has become the populus trichocarpa reference genome. It's a great resource. But by the time we got to 2016, I sequenced the genome of 1,000 poplar trees, and each of them cost less than $1,000. What this means for breeding is that we can sequence every individual in our populations for breeding, and it means we can be using the data from the genome to inform the pipeline in a different way than had been used previously. And as I say, I'm not an expert in this, but I do have some wonderful colleagues who are experts. But this genomic selection really relies on using a training population, which is shown here, which is both phenotyped and genotyped. And there are various ways of developing a training population. We have one in poplar, for example, at the moment, which is a very complicated set of crosses. And this population is used to train a model. So the difference then is that we're not trying to identify a single marker and associate it to a single trait, because that hasn't been very successful. We're using markers across the whole genome. So it's an improved form of this business we call marker assisted selection. So it aims to capture the small effect genes which we know are important for quantitative traits. So you have this high density of markers, and you have the training population, which is both phenotyped and genotyped. And this information is used to select from the breeding population, where this population is only genotyped. So we only require genotypic information to make predictions on how we should move the breeding program forward. What this means is that genotyping information is very important because it determines the model to some extent, but also phenotyping now has become key to developing these accurate models. And phenotyping is relatively expensive, and it's where our current focus needs to be to address this challenge of high nutrition crops. So these were some ideas given to me by Dan Runcie, so please contact Dan if you'd like further information. So essentially, if we have this pipeline for breeding and we're making crosses and trying to get commercial release, what genomic selection and AI can help us to do is accelerate the pipeline by enabling us to make predictions more quickly. So we can make these predictions without phenotyping everything, but we need very accurate phenotyping in our test population. Now, there are problems with this. There are multiple ways to model the way this genomic selection. And it turns out that they're very tightly contained in the genetic background in which the populations are being studied, in which these models are being developed. And so we don't have all of the answers. And so there's a lot of complexity here that we still need to understand. But as I've said, phenotyping is really important, and accurate phenotyping. And I just wanted to finish by a project that we're actually doing within the AI Institute for Food Systems. This is being led by Christine Diepenbrock, and she gave me these slides in collaboration with Charlie, Brian Bailey in Plant Sciences, Mason Earls from Viticulture and Enology, Dan and Troy Magny from Plant Sciences, and Johnny and Earl are the two primary researchers, with some additional collaborators, including Duke Pauly from the Arizona phenotyping rig, which we've used to collect our lettuce data. So the overall approach here is to use AI to unravel some of these complexities that I have been describing in this talk. And it's really here to get a better handle on remote sensing observations, in situ measurements of leaf quality and nutrition, vigor and yield traits. So we're interested in multiple horticultural crops. We're interested in beans, lettuce and spinach, alfalfa, and also strawberries. So these are high value crops where we'd like to improve nutrition. And so we can take spectral, hyperspectral data gathered in the field. We can try to model these data in different ways, so we can put this into our genomic prediction, our breeding pipeline, and we can use multiple traits simultaneously in the future. So that begins to get us at these unknowns for nutrition. And we can get better phenotypes to breed for nutritional crops in the future. And this is very much cutting edge research. It's what we're doing. So we use hyperspectral data to get us our phenotypes. We're combining this with genomic prediction, our genome data and crop models to improve breeding in future. So currently, the emphasis in this year is on testing the use of sensing data and using the modeling approach here that Christine and co-workers are using is this convolutional neural networks approach, so very novel, to predict the traits that we want to use in breeding. And it's to incorporate both these proximal and remote sensing data to our ground truth data into these modeling frameworks, and to interface these with our high resolution, those genetic SNPs data, which I also described. So that's really where we're at with this type of research. And as I said, we're looking at lettuce, spinach and alfalfa and common beans. And you can see here, this is the setup of Troy Magny's lab. It's mounted on an 80-foot tower. It's solar-induced fluorescence and infrared and an RGB camera where we can get at multiple traits using this sensing that sits over a bean crop here. And in contrast to that, this is the gantry in Arizona. So we have a diversity panel here in collaboration with Duke and Richard Mitchellmore. The gantry scans several times an hour. I think it's every 11 minutes for all sorts of traits, remote traits, which we're now trying to gather together to figure out how we should move forward. It collects about 1 terabyte of data a day, and it's quite fun. I've worked in this site myself, and this is an infrared image of our mapping populations of lettuce and how they look in Arizona. This was the very last thing we did. It was about the 9th of March, 2020, just before the pandemic. So we're still looking at these data and figuring out what they mean. So that's a little bit of insight into what we're doing currently. So I hope I've addressed these 3 challenges. Crop nutrition and quality haven't traditionally been a focus. They're coming to the fore now. Traditional plant breeding is slow, but we can address it. And we must wrestle with these complexities the whole time. The thing I haven't talked about today is this social license to operate. I don't feel I have the expertise. I have some insight into how things are going, but I think it's something that we do need to think about the whole time. So what can we conclude? So there's a pressing need for this accelerated breeding for plant nutrition. We have global health targets. You look at the very poorest nations, the African continent on one hand, that we need to get more highly nutritious food developed. And on the other hand, those searching for the promise of nutrigenomics, nutrigenetics, and personal health, which will also come along in the next decade. So AI offers significant potential here to accelerate the breeding pipelines for predictive modeling, targeted and improved phenotyping and genomic breeding. One of the biggest challenges at the moment is deploying phenotyping at scale with improved sensors and cheaper sensors and cheaper approaches. These are still pretty big, expensive, time-consuming experiments. And then in the nutritional world, really, what are the unknowns? How can we find them? How can we work together better across this spectrum of health, plants' health, and figure out what we really should we be breeding for? Thank you very much for your attention. That's all I have today. Thank you.
Thank you so much, Gail, for that illuminating presentation on the challenges and hints at the potential solutions to these challenges in molecular breeding. I would encourage everyone to submit questions in the Q&A section, and we will go through those. So I'll give you a second to type those out. And I can start us off with a question. I know that you don't want to get too much into depth on SLO, but if you could maybe illuminate us a little bit more on that topic.
Well, I guess the social license to operate, the world's a little bit different. I have a map actually of the world, what the regulatory environment looks like. And at the moment for gene editing, there's a very big difference between what's happening in the USA and what's happening in Europe, what's happening in the UK even. So Europe is more or less still remaining on the thesis that gene editing should be treated as a mutagenic type of breeding, and that it really isn't accepted by the public. Whereas in the US, the US has taken more of a functional approach. That's not to say it's entirely accepted by the public, but it seems that there's more willingness to accept. And then the rest of the world is moving, from my perception, with some exceptions such as New Zealand, seems to be moving towards the US-Canadian stance. So, but my fear is that with the sort of multiple marker genome enhanced breeding, something will happen that upsets the public, and it could all go horribly wrong, and we may end up in some terrible place that we shouldn't be. And so we need to be addressing that before that might happen. So I think it needs a lot more thought. I'm not necessarily the best person to do it, but I think that we've seen the mistakes we made with GMOs, that some public acceptance was lost there, and sort of science was seen as the bad guy. So I do think we need to think about that a little bit more.
Yes, building trust is always an important part of the relationship between science and public perception. Let's see, we have a question in the chat here. Can you elaborate a bit more on what are some of the phenotyping features you look for, and how are they different across plants?
So we're working on a variety of characteristics. We're using quite a lot of hyperspectral imaging where we can elucidate some of the important chemistries for health for humans. So pigments, the vitamins, so for example, vitamin A we would look at routinely, chlorophyll and NDVI, so greenness. We look at the anthocyanins, so redness of food types. Anthocyanins are also linked to health-giving benefits. So that's those. And then we're also working on traits which are more about resilience and input, so climate sustainable crops. So, for example, we're working on infrared images for reduced water use. So crops which can use water very, very efficiently. I guess going back to those hyperspectral and nutrient characteristics, I just feel we're just scratching the surface there, that we need better sensors. In another project, I work on some open source, ultra cheap sensors for measuring other things. And I think there's vast potential there to do more. It's getting at these unknowns. So we need more insight into what we should be measuring in plants and how we can get at that really, really quickly.
Yeah, makes sense. And then this next question, can you speak to our community in the Sacramento and Davis area and the seed breeding operations that are here, such as Unfold, Bayer, Sakata, and others? Do you see AIFS engaging in a deeper way with these entities and others through the research that you described today?
I mean, we do interact very, very largely with the local. I mean, it's fantastic. This area is often called Silicon Valley for seeds, and we have multiple complex interactions with the local companies. So we train the people that go to work in the companies. We have collaborative research projects. We have something called Seed Central, which is run by Seed Biotechnology Center. So that the director there is Alan Van Dyne. We meet regularly. We meet monthly. It used to be a fantastic networking event where we all got together. Obviously, we haven't been doing that for a year and a half, but hopefully we will get back to that. So we're very embedded with those communities, but we can always do more. So I'm always really interested to hear how AIFS in particular can engage our breeding with those local companies. So yeah, we have our advisory board within the Institute, and they have some local representatives, but rarely a week goes by where we don't have some contact with our local community of commercial companies who are working on plants. I'm very often, as I know my colleagues are, out in that environment. But if there's any way we can do more, that would be fantastic. I guess it goes back to this innovation, that bringing together mathematicians, sensor developers, digital people, and plant people. What we need to be doing in the university is really innovating in that domain. So we can think of the companies as pushing things through breeding pipelines to get varieties out really quickly for commerce, for commercial success. But the university is maybe one step removed from that, and we want to get these innovations together so that we're developing things that are not off the shelf. So there is a sort of a technology pipeline, and we both have a role to play. But yes, if there's ever any way we can improve, I'm always interested to hear.
Fantastic. Yeah. Always good to hear about the connections there and applications for the research being done. And then this next question is in regards to the orphan crop part of your talk. How have African consumers and farmers responded to the work of the AOCC? Has there been any pushback?
Not to my knowledge. I mean, Alan Van Dyne is the expert, but the amazing thing that I feel that they have done is that they have the African crop breeding academy. So and they are training the next generation of breeders who will be based in Africa, breeding those crops that are not maybe of global significance, although they're very interesting to us, to work in their particular environments, to take those crops forward and to improve them. And that can have an amazing knock-on effect for the welfare, for the commercial success, for the livelihoods of those local communities. So I hope it's a really positive thing. And I haven't heard of any pushback. I've only heard really good, amazing, positive reports.
Outstanding. That's good to hear. See any other questions out there. Okay, here's one more. Can you comment on applying AI to find the needed healthy nutrient components for humans from plants versus AI for plant breeding?
Gosh, that's a tough one. I don't think I can. Really, I think is the question asking how we link up those two very disparate bits. So I might be able to breed a really interesting, nutritious crop with my colleagues, and let's imagine we can accelerate that through the pipeline. In a couple of years, we have something really interesting. The question then is how that impacts human health, and that's that's making claims about diet and health, as we heard in our last talk, has become incredibly difficult, particularly in Europe, but also in the US, making claims and proving them. And so I'm sure there is a role that we can unravel, just the microbiome of humans, how it's impacted by plants, and how we make that pipeline work faster. I don't have the answers, but I know it's where the subject needs to be. And there are a couple of institutes, and I know some of the big companies have invested hugely in bringing those domains together, and we've heard some of that. I don't think I've seen any real outputs yet, real significant commercial outputs. So it's still a work in progress, but I'd love to talk about it more.
Yeah, and I think that's an excellent question for the panel that we'll have on December 8th, where we gather the experts from our speaker series to have more of a panel discussion and dialogue on these sorts of topics. And let's see, this is a long question here. Following up on the earlier question about the African Orphan Crop Consortium, one of the reasons that we got where we are in terms of the reduced number of crop varieties that are available and the subsequent reduction in nutrition and variety in our diet is that breeding has traditionally been driven by companies whose only goal is profit making, regardless of the negative effects on the food ecosystem. When I look at some of these consortia, such as African Orphan Crop Consortium, I become skeptical of their goals simply by looking at who's involved. For example, all the organizations involved are Western for-profit organizations, and there's not a single African university or national research institute listed on their website as part of the consortium. The leadership of the consortium is Western without a single African decision-making person. Do you think that with AI and other technology, we are going to achieve the goals we hope to achieve if there are no representatives of the people these new movements are targeting? How is their voice represented?
Gosh, that's a big question. But you make several really, really excellent points, and it's a great question. You are correct that we are where we are with breeding in the world because a lot of breeding has been in the hands of some big companies, and there have been lots of sort of consolidations. But don't equate profitability with something being bad. What those companies have done is a fabulous job in the sense that they've given us some food at an incredibly cheap price, that the cost of food is relatively very low at the moment. So they did do a job. We all benefit from that. But there is still, I'd still have a shout out for public breeding, plant breeding programs. UCDAVIS, Charlie Brum is the director of the Centre for Plant Breeding at UCDAVIS, and we do still make public releases. So we have a student program on breeding where they're releasing varieties in several crops and flowers. So public breeding does still go on. It's important. And also universities do a lot of pre-breeding. So they make germplasm, they make advanced lines, which they then hand over to anyone, sometimes unlicensed, so that the best benefit can be made from those. But I totally agree. I'd like to see more diversity and you know, that's what we, a lot of university people feel that way, that we need to sort of address that. So let's take some action and make that happen. And the way I see the African Orphan Crop Consortium is a small step to try to do that, but it doesn't achieve everything. It's doing something, which is better than doing nothing, but I think it could still do a huge amount more. Hopefully, if it could grow into the next phase, there could be more on the boots on the ground in Africa that could enable that to happen. And maybe if we're thinking about AI and how we deploy it, we should think about that, and we should think about those equity issues. You know, it sounds like the sorts of thing that Gates Foundation might be interested in funding and taking it to the next stage. But I think we have to try and move forward in a positive way. There was an amazing article, I think it was the New York Times, about how sort of seed reserves and germplasm has been reduced over the past decades. And we know it's really important, but we're here to stop that and to do better, I think, in the future.
Great, thank you for your answer to that excellent question. We're coming up on time here, so I guess the last question that we can answer here is how people can engage with AIFS. And if you want me to take that one, please. I would be happy to. So you can engage, first of all, by continuing to come to our speaker series. We have 2 more this fall, and that will be on next Wednesday. Let me pull this screen up again. Next Wednesday, how AI can improve food safety, helping to enable safe food for all, from Dr. Abigail Stevenson at Mars, and then the speaker panel that I mentioned earlier. If you would like to connect with AIFS, you can email us at aifs@ucdavis.edu. If you're a part of the world of industry, we have a business development coordinator, Jim Pantaleo, who would be happy to discuss more with you about how to connect that way. And I'm also available to connect with as the public engagement and education coordinator. So thank you, everyone, for coming today. And thank you so much, Gail, for speaking with us. This has been a great talk. And we really appreciate you taking the time to discuss with us.